Welcome to our channel DS Institute of Bioscience. In this channel, we will gonna talking about class 9, class 10, class 11, class 12 life science related topics. So today's topic of discussion is the photosynthesis light dependent reaction. Now, what is the photosynthesis? The photosynthesis is the photochemical, biochemical, anabolic oxidoreductive process by which the green plant cell can prepare carbohydrate food material, mainly the glucose, in presence of sunlight, water, carbon dioxide, and chlorophyll, and producing O2 as the byproduct. As you've seen in the diagram that the carbon dioxide and water with the help of chlorophyll and the sunlight get converted into the glucose, water, and producing O2 as a byproduct. Now the question is the why the photosynthesis is called the photochemical reaction, because this reaction occurred in presence of light, mainly the sunlight. That's why it is called the photochemical reaction. So now why it is called the anabolic process? Because in this process the glucose is synthesized, so the dry weight is increased. That's why it is called the anabolic process. So now why it is called the oxidoreductive process? Because in this reaction the carbon dioxide is reduced to form the glucose and water is oxidized to form the oxygen. That's why it is called the oxidoreductive process. So next is the side of photosynthesis, where actually the photosynthesis takes place. The photosynthesis takes place within the chloroplast containing the mesophyll tissue, that is the palisade parenchyma cell and spongy parenchyma cell present between the upper and lower epidermis of dorsiventral leaf. As you've seen in the diagram that water molecule is moved out from the xylem vessels and absorbed into the palisade parenchyma cells, that is also involved or take part in the photosynthesis. So next is the other side of photosynthesis. Some bacteria can also perform the photosynthesis, such as the cyanobacteria and the purple sulfur bacteria. And epiphytic roots of orchid, it is the example where the root can perform the photosynthesis. That is called the velamel, and it can also absorb the atmospheric moisture. And these epiphytic roots of orchid can perform the photosynthesis. So next is the phase of photosynthesis. The photosynthesis phase actually broadly classified into two main phases. One is the light dependent phase, that is also called the Hill reaction, and another is the light independent phase, that is also called the Dark reaction or the Kelvin cycle. So next is the light dependent phase. The light dependent phase is the phase of photosynthesis that we cannot perform without the presence of light. In this phase, a reductive reaction takes place that causes the formation of NADPH, that is further used in the dark reaction and also produce the O2, H2O, that is the water, and ATP. Before I starting the light reaction in details, there are some aspects regarding the light dependent reaction that we have to know. So first is the spectrum. Spectrum are classified into two types: action spectrum and the absorption spectrum. Absorption spectrum describes the wavelength of light that is actually absorbed by the green leaf, and the action spectrum is the wavelength of light that actually drives the photosynthesis. Plants are actually absorb the red and the blue light and reflect the green and the yellow light. That's why the leaf looks like the yellow or green in color. Sometimes it is also asked in your exam that's why the leaf looks like green or yellow in color. The reason behind that is the same. Another aspect of the light dependent phase is the pigment system. In the light harvesting complex, there are two types of pigment system is present. One is the PS2 that activate in 680 nanometer of wavelength of light, and another is the PS1 that activate in 700 nanometer of wavelength of light. For your additional information, the PS2 is blocked by a chemical that is called TCMU, 3,4-dichlorophenoate phenyl 1,1-dimethyl urea, and PS1 is also blocked by another chemical is called the paraquat. After absorbing the sunlight, the light harvesting system transfers the electron in two ways. One is the non-cyclic electron flow, another is the cyclic electron flow. And the choosing the non-cyclic electron flow and the cyclic electron flow is completely depend on activation of PS1 and PS2. If the light intensity is more suitable for PS1, then it choose the cyclic electron flow. But if the light intensity is suitable for both PS1 and PS2, then it choose the non-cyclic electron flow. So let's start with the non-cyclic electron flow. In the non-cyclic electron flow, the electron transport chain, that is the ETC, it is consist of the electron acceptor and the electron donor is located on the thylakoid membrane. So at the beginning, when the sunlight, mainly the 680 nanometer for the PS2 and the 700 nanometer for PS1 hits the PS1 and PS2 simultaneously, that causes excitation of chlorophyll of both of them and lead to release of electron from both of them. And the electron from PS1 move through a iron sulfur cluster to the ferredoxin, that is the FG, and the ferredoxin then transfer the electron to the ferredoxin NADP plus oxidoreductase, that is the FNR, and this enzyme then transfer the electron to the NADP plus to convert it NADPH. Now the PS1 chlorophyll have a deficiency of electron. On the other hand, the electron from PS2 move to the plastoquinone, that is the PQ. As because the plastoquinone received the electron, it gets reduced to from the plastoquinol, that is the PQH2, and it transferred the electron to the cytochrome B6F. And after releasing the electron, the plastoquinone again oxidized to from the plastoquinone. And the cytochrome B6F then transfer the electron to the plastocyanin and import 2H plus from the stoma into the thylakoid lumen. Plastocyanin then transfer the electron to the electron deficit chlorophyll of PS1 to normalized state. Now, as because the H plus ion is pumped into the thylakoid lumen, that's why the H plus ion concentration is more inside the thylakoid lumen than the stoma. So the H plus ion get pumped out down this concentration gradient into the stoma by the ATP synthase, and it causes formation of ATP from the ADP and PI. But there is a still electron deficiency present in the chlorophyll of PS2, and that can be resolved by oxygen evolving complex, that is the OEC. In the OEC, there is a manganese containing protein is present, and the sequential change from S0 to S1, S1 to S2, S2 to S3, S3 to S4, in each change one electron is released, that is accepted by tyrosine YZ of D1 protein. And upon receiving 4 electron, 1 electron is transferred to the PS2 to normalize it. And during that situation, oxygen evolving complex causes breakdown of water, and it is called photolysis of water to form the O2 and H plus ion, and that causes evolving of O2. So the evolving of O2 is occur from breakdown of H2O. So all the point that I described in my previous slide is written here. You can pause the video and make your own note. So next is the cyclic electron flow. In case of cyclic electron flow, the sunlight, mainly the 700 nanometer, hits the PS1, and that leads to excitation of chlorophyll of PS1, and that causes release of electron from PS1. And that electron moves through the iron sulfur cluster, and then the iron sulfur cluster transfers the electron to the ferredoxin, and ferredoxin then transfers the electron to the cytochrome B6F, and cytochrome B6F transfer the electron to the plastocyanin and import 2H plus ion from the stoma into the thylakoid lumen. Plastocyanin then again transfer the electron to the PS1 to normalize state. So now the H plus ion concentration is increased inside the thylakoid lumen. So the H plus ion get pumped out to down its concentration gradient into the stoma by the ATP synthase, and that causes formation of ATP from ADP and PI. All the points that I described in my previous slide is written here. You can pause the video and make your own notes. For your additional information, the electron flow through the electron transport chain is due to difference in redox or reduction potential of electron carrier in the electron transport chain. Electron always moves from the lower reduction potential to the higher reduction potential. That means in the ETC, there is a presence of electron carrier, and the electron carrier are either electron acceptor or electron donor. So the reduction potential of electron donor is always lower than the reduction potential of electron acceptor. That's why the electron flow in a particular direction in the ETC. So that's all about light dependent phase of photosynthesis. I hope you enjoyed this video, and if this video was helpful, please subscribe our channel and share with your friends. Thank you.